So we now know that after the fluctuation at the presynaptic neuron near the receptors of the fluctuation of the currents both by excitatory and the inhibitory, finally by summation of those positives and the negatives, the action potential finally starts. If it can raise the membrane potential maybe 15 millivolt higher than 70 millivolt, so that the firing need to reach the firing level at minus 55 millivolt. Now the action potential, with the action potential, the current reaches finally at the final presynaptic membrane of the first order neuron, because there is a synapse between the first order neuron and the second order neuron at the level of dorsal root ganglion. So this synapse, near the synapse, when the first order neuron ends, and the second order neuron starts. This is the synapse. And near the synapse, to keep these two neurons together, there are some special type of cells we have discussed already. Those are astrocytes. So these astrocytes act as scaffolds of these two neurons, first order neuron and second order neuron, so that they can be kept together. We also discussed that with the astrocytes, they make tight junction with the blood vessels to receive the glucose and also the oxygen necessary for production of energy or ATP. So what happens when the action potential reaches, that means the minus 70 millivolt which was here, that becomes plus 40 millivolt. So when one after another the sodium enters, the sodium goes on entering. Finally, when the sodium enters here, and what happens, and how the second order it reaches here, the action potential reaches up to this level, the end of the first order neuron. Now this current cannot directly pass on to second order neuron. So you need some chemical messenger. So this current or electric energy has to be again transducted into chemical energy. So there is also another transducer at the end of the first order neuron, that is a little bit thickened or active zone of the first order neuron, this presynaptic membrane. Similarly, the beginning of the second order neuron, that is also active, and so that is also thickened, and it is seen in the electron microscope as a dark bands. So this is also an active zone, this is also active zone, because lots of transductions occur here only. How? We know that there are some clear vesicles. These clear vesicles, they receive the neurotransmitters. The neurotransmitters, there are many neurotransmitters. Maybe for our discussion, we know that there is glutamates. See, these are glutamates. Glutamates are produced from an amine, that is glutamine. So after the glutamates are produced, it comes to the microtubules from the nerve cells, and these glutamates are here, and the empty these clear vesicles which are produced by the budding of the endosomes. They receive and they are filled up by the neurotransmitters. After they are filled up by these neurotransmitters, these clear vesicles are translocated. This translocation done, and this translocated clear vesicles, they still contain those glutamates inside those. Now, these glutamates, these clear vesicle, they will be moved towards the first order, the presynaptic membrane, and this movement is typically, it is called as docking. So it is docked. So this docking is done, and the docking is done by some bonds, and these bonds are, there are two types of bonds. These are synaptobrevin and syntaxin. So synaptobrevin and syntaxin. These two bonds, they keep the vesicles together tightly with this active zone of the presynaptic membrane. And inside there are already the glutamates. Now it is primed with ATP. After it is primed, so now the priming with the ATP, it is now ready to liberate the neurotransmitters, but it cannot release the neurotransmitters unless the calcium enters. So when the first order neuron, the electric current reaches the terminal, then it is ready for entry of the calcium. So the calcium ions which were moving around in the synaptic space and on the extracellular fluid, now they will enter. And if the calcium enters, immediately there will be exocytosis. So the exocytosis of the glutamates will occur. So these will come out, and these will be there in this synapse. This, after exocytosis, then again these clear vesicles, there is endocytosis, and it closes. The opening of this clear vesicle is closed. So the endocytosis starts, and then the opening is closed, and these clear vesicles again moved inward by the process of the translocation, and finally it fuses with the endosomes, and the endosomes again budding occurs, and then again neurotransmitter. So this is a cycle of the glutamate release from the first order neuron. We must know that what the purpose of these glutamates. We know in the postsynaptic membrane there are also some pores, there are some small doors, or these are called AMPA receptors. Small doors are called AMPA receptor, and there are some big doors which are called NMDA receptor. Normally, the AMPA receptors can accommodate the sodium ions. They enter into the AMPA receptor after it is activated by these glutamates. So if the glutamate is attached to the AMPA receptor, it opens, and this sodium goes inside. And once the sodium goes inside, this membrane potential is further changed, and it becomes positive. So another action potential is generated, and so the sodium enters through this AMPA receptor after the glutamates are attached here. Similarly, in the NMDA receptors also, the glutamates are attached, and the NMDA, so the glutamates are excitatory, glutamates are facilitatory. It initiates the action potential. There are also some inhibitory neurotransmitters, that is, these are also liberated. GABA, by the decarboxylation of the glutamates, GABA is produced. Glycine, Glycine is also inhibitory. So glycine, GABA, these are also there. And this glycine, but remember, glycine is excitatory at the NMDA receptor. Normally glycine is inhibitory everywhere, but at the NMDA receptor it is excitatory. So these amino acids can be, glycine can be both excitatory and also inhibitory. But glutamate is definitely, it is excitatory, and GABA, it is gamma aminobutyric acid, that is definitely an inhibitory amino acid. Now, these glutamates, after these sodium enters, what happens? If there is continuous inflammation, the ongoing pain, if the, say for example after a surgery, an analgesia has not been given adequately, the patient is having pain, that means continuously the action potential is coming. Continuously these glutamates are being released. So lots of glutamates are there in the synaptic membrane, and the glutamates almost stimulated all the AMPA receptors, these ion channels, and so sodium has entered all through. So this membrane potential remained a little up, and the threshold has been reduced considerably. And if the threshold has been reduced considerably, then at the NMDA, if both these glutamates and also the glycine are attached, then the NMDA receptor is activated. Normally, this NMDA receptor is blocked with a big magnesium. So magnesium ion blocks like a doorkeeper. But if this NMDA receptor is activated by both glutamate and glycine, then calcium, which is there, this calcium will dislodge this magnesium ion. This magnesium ion will be dislodged, and calcium will enter, and this calcium will enter in. After that, you remember that this calcium, when it goes inside the cells, something happens. We have seen the release of the glutamates, and we have also seen other things like that, the calcium now entering in the postsynaptic membrane. Then what happens? This calcium is attached with there is a protein which is attached to calcium, that is calmodulin. So calcium and calmodulin, Ca plus Cam, so calmodulin, there is a compound is prepared, and this calcium calmodulin, now it is attached with the kinase 2, and this kinase 2 means calcium calmodulin kinase 2. This compound or this complex, what they do? There are also vesicles. There are also vesicles in here. They also produce proteins. So, with the influence of this calcium, calmodulin, and kinase 2, there is phosphorylation, and AMPA receptors are prepared. These AMPA receptors then are translocated, and in between, so newer AMPA receptors are there, and this also phosphorylates this AMPA receptor. That means it increases the sensitivity for these glutamates and other things. So as a result, there is a vicious cycle. The membrane now becomes more responsive, more sensitive, and also there are other happenings. There is a gas, nitric oxide, from the arginine amino acid is produced. This nitric oxide diffuses. It diffuses through the synapse to the first order neuron. And this nitric oxide, they actually stimulates to produce more glutamates. So more glutamates are produced, and also they stimulate to release substance P. The substance P is normally lowers the threshold of the first order neuron further. And this substance P and the glutamates, as it is more, as we have also seen that this glutamate actually is taken up by the astrocytes. So this substance P and glutamates both are taken up into the astrocytes, and in the astrocytes, when there is more glutamates and more substance P, this astrocyte will liberate C-phos and also BDNF, brain derived nerve factor, and also NK1, neurokine 1. So these are also created by many cells, including these astrocytes. These interleukins, they also stimulate this interleukin 1 beta, which again goes to the presynaptic membrane, and this interleukin will again produce more glutamates. So there is a vicious cycle, and this C-phos, this oncogene, it produces growth of the nerves. So it becomes wider area, wider sensitive area, and this wider hypersensitivity at the level of the spinal cord by ongoing pain, and the activation of the NMDA, recruitment of this more and production of more facilitated neurotransmitters. This as a whole is called long-term potentiation or potentiation. So this long-term potentiation, it is found mostly at the level of the studies have been done at the level of hippocampus, but it occurs at the level of the spinal cord, at the level of the dorsal horn cells.